Hello, everyone, and welcome to this new Gen webinar entitled Advancing Direct Enzyme Delivery for Therapeutic Genome Editing in Vivo. Today's webinar was made possible through joint sponsorship from Aldevron and Synthico. I'll be your host for today's presentation. I'm Jeff Bogaliskis, the technical editor for Genetic Engineering and Biotechnology News, which is in its 40th anniversary year, covering all aspects of the life sciences industry. Undoubtedly, one of the hottest topics that Gen has covered in that time frame has been genome editing and the therapeutic potential that it holds. In particular, ex vivo approaches have demonstrated this promise through the use of gene editing enzymes such as CRISPR-Cas9, which have been successfully employed to target different genetic disorders in model systems. Yet the challenge of delivering these curative enzymes in vivo is still a large clinical hurdle. As such, investigators have turned their focus to viral vectors, which now represent the most widely used delivery platform in preclinical work, with nanoparticles quickly maturing into an equivalent transport vehicle. Direct delivery of preformed CRISPR enzymes has recently emerged as an appealing strategy for enabling therapeutic editing in vivo, and something our presenters are going to tell us more about in just a few moments. But before they do, let's meet our three speakers for today's event. Up first will be Dr. Ross Wilson, an assistant adjunct professor in the Innovative Genomics Institute at the University of California, Berkeley, where his lab focuses on enabling the widespread clinical use of genome editing enzymes. Ross and his team utilize protein and RNA engineering to create safe, effective methods of administration that will enable targeted delivery to specific cells, tissues, and organs, something he's going to tell us a lot more about in his presentation today. Next, we'll turn the presentation over to Max Selman, a product manager at Aldevron, where he focuses on gene editing protein products and customer service. Today, Max is going to tell us about the company's custom GMP ribonucleoprotein production platform. Finally, bringing the presentation to a close, we're going to hear from Dr. Kevin Holden, who is the head of science at Synthico, where he is a senior member of a research team responsible for integrating CRISPR genome engineering workflows into novel automation platforms. Today, Kevin is going to wrap things up by telling us how Synthico has been helping to support the possibility of in vivo CRISPR therapeutics through its RNA synthesis capabilities. But before we begin, I want to remind everyone to stick around for the Q&A that will begin right after the presentations. Don't be shy. Send in any question you have at any time during the presentations for any of our speakers. It's really simple. All you need to do is click that Ask a Question tab right there on the right-hand side of your screen, type in your question, and hit Submit. Like I said, you can do that at any time during the presentation, so don't be shy. Send them in as early as possible so we can try to get to as many of your questions as possible. Okay, now that we all know how to ask questions, hopefully everyone will send in a bunch for our panel. Let's get things started and open the floor up to our first presenter. Ross?
Welcome, and thanks for attending this seminar. Today, I'm excited to be telling you about some recent advances in the therapeutic use of genome editing enzymes, as well as efforts by my lab and others to improve delivery of these preformed CRISPR enzymes for therapeutic use, ultimately in vivo. My name is Ross Wilson. I'm an assistant adjunct professor at UC Berkeley, and I'm the director of therapeutic delivery with the Innovative Genomics Institute. So if you're here, you probably already have some interest in genome editing. There's so much excitement and promise in the realm of genome editing, especially when it comes to therapeutics. We could spend many hours just talking about all the promising preclinical evidence that these enzymes have the power to turn off malfunctional proteins or genes or restore gene function. I just want to highlight that in addition to the standard CRISPR function of creating a double-stranded break and allowing one of these two pathways to take place, recent developments have created base editors and prime editors. And these modified CRISPR enzymes are able to restore the function of a gene or disable the function of that gene without creating a double-stranded break. And these enzymes have a lot of potential, and they also have special considerations when it comes to delivery. So I want to highlight this before we begin. So when we talk about CRISPR enzymes, we're always talking about RNPs. RNPs are ribonucleoproteins, or RNA protein complexes. These consist of a Cas protein, which is typically going to be the nuclease, which creates that double-stranded break, and it can bind to its guide RNA. So the guide RNA has a constant region, which acts as sort of a scaffold, and this is attached to a programmable region, which lets you send your homing CRISPR enzyme to any part of the genome that you want. So we're going to be talking about delivery and therapeutic use of preformed CRISPR enzymes today. And this means I'm going to be saying RNP about 1 million times. So whenever you hear RNP, think about a preformed enzyme that's ready to go. So we are all aware of the massive therapeutic promise of CRISPR-driven genome editing, but the impact in the clinic has been relatively limited. And this is because we lack delivery options. So one great way to get CRISPR into the clinic is to sort of avoid the delivery challenge entirely and just take the cells out of the patient and put them in the lab where they can be easily manipulated. And CRISPR has already been useful in the clinic in this regard, and this is analogous to earlier gene therapy or cell therapy approaches where a viral vector is used to deliver the genetic cargo, and it could be even used to deliver a CRISPR payload. So ultimately, the entire field would love to move towards in vivo delivery, but this is a big challenge for some reasons that we'll talk about today. And there is a lot of hope in various realms when it comes to different platforms that can be used for in vivo delivery. And these sort of span a spectrum from established technologies like viral vectors, which were initially developed for traditional gene therapy. And then a sort of intermediate type of platform is nanoparticles. And these historically were developed for delivery of therapeutic oligos like ASOs or siRNAs. And a lot of my personal excitement is around the delivery of preformed Cas9 enzymes or CRISPR enzymes like RNPs. And I think this has a lot of potential, but it's a relatively new field, so it's still very much developing. So all of these different approaches have pros and cons. This slide introduces a sort of hybrid between nanoparticle and RNP strategies where a nanoparticle is used to convey preformed RNPs, and we're going to talk about some published examples of this strategy. But I don't want to take the time to go through every detail on this slide. Essentially, viral vectors look great in preclinical data, but they have not had a great translational track record. And I think this is because they can be associated with genomic integrations or increased off-target cutting because of the prolonged expression of the genome editing payload. They can also induce immunogenicity. So the host will respond negatively to both the viral vector and overexpressed cargo. And it's very, very hard to manufacture large amounts of viral vector. So if a mouse is dosed with a huge amount of viral vector, a human patient probably can't be dosed simply because it's too hard to make. So those are some of the downsides about viral vectors that have limited their translation despite lots of really encouraging preclinical data. Lipid nanoparticles are great. These will carry an mRNA transcript of the Cas9 along with the pre-made guide RNA. Intellia is actually taking lipid nanoparticle delivery of Cas9 mRNA to the clinic to target the liver. And this is where nanoparticles really shine because the liver tends to filter these particles. They're a little bit bigger, and we're going to talk about the size of these particles subsequently. As far as in vivo distribution goes, nanoparticles carrying RNPs are quite similar in that they're larger, so they're sort of limited in how they can be distributed, but they've seen a lot of promise via kind of local administration. So we'll talk about some cases where the ear, for example, the surface of the ear or the surface of the lung could be edited by a preformed editor carried by some sort of nanoparticle or peptide complex. And so finally, the thing that my lab works on that I'm really excited about is actually engineering the RNP enzymes themselves to give them all the properties that one might want for targeted delivery in vivo. And one reason that this could be really appealing is that these are smaller particles, and it sort of opens the door to improved biodistribution. So just to take you through, before we move on, I should really highlight the different sizes. RNPs are the smallest. AAV is the kind of standard or most popular viral vector, and it's a little bit larger, and nanoparticles are typically at least 50 nanometers, and many of them are as big as 100 nanometers. So just to get you all really excited for all the virtues of working with engineered RNPs for therapeutic delivery, we'll do the sort of sales pitch for this platform. It's really appealing because there's no overexpression. The preformed enzyme is transient. It only exists inside the targeted cell for 12 or 24 hours. This means that there will be decreased immune responses and fewer off-target effects. And this is a big advantage, especially compared to viral delivery. The RNP is made out of recombinant components. So the two other speakers on the call with me today represent companies that will produce for you either guide RNAs or CRISPR proteins. This is a huge advantage because scale up of these materials works really, really well. And this is in stark contrast to the challenges of scaling up a viral manufacture. And not only is this great for research purposes, but it should work great in a clinical context where you need essentially massive amounts of these preformed enzymes. So the Cas enzyme itself is not able to enter a cell without any modifications. This is an apparent disadvantage, but we hope we can turn it into an advantage, because if it has no inherent ability to enter any cells, it should only be targeting and entering the cells that we engineer it to target and enter. So we'll talk a little bit more about that when we get to my lab's work in the latter half of the talk. The fact that RNPs are small should give us improved distribution in vivo, as I mentioned earlier. And another really exciting thing that gives this approach an advantage over viral vectors is that there's no packaging limit. So the adeno-associated virus, AAV, has an upper limit of about 5 kb of genetic cargo that it can package. This makes it borderline impossible to package something like a base editor or a prime editor. However, with a preformed Cas enzyme, there's no inherent limit to how much can be packaged or assembled for your delivery project. So adding an extra domain isn't going to dramatically change the properties of your delivery approach. So with that, you should be primed for the RNP takeover of therapeutic genome editing. And now we're going to get into the first of two main chunks of my talk. First, we're going to talk about where we are. And this will include published work that I think is really exciting. And in the second half, we're going to get into where we're going, which is some of my lab's unpublished work. So engineered T cells have been therapeutically useful for a really long time. And CRISPR-Cas9 has made a big splash in advancing and improving the ways that these cells can be engineered. So historically, a CAR T cell would be created by taking a patient's T cells and adding a lentivirus that can insert a CAR gene to add this sort of orange engineered T cell receptor, for example, on the surface. However, without the ability to remove a lot of the other cell surface markers, this won't be an optimized engineered cell. So Carl June actually took CRISPR engineered T cells to the clinic. And in doing so, they did attempted a triple knockout of these T cells to remove the endogenous T cell receptor, as well as the therapeutically important protein PD-1. And in doing so, they were able to remove all of these cell surface markers that they don't want present in their engineered cells. So they used CRISPR to do this. Unfortunately, this was a pretty early study, and it didn't look like too much efficacy was imparted. And one of the reasons is because they found it difficult to do multiplexed editing with 3 cutting Cas9s. And so subsequent work that I'll tell you about soon actually found a fix for this. Other work has also found a way to remove the need for lentivirus entirely. So some work from the Bush and Marson labs showed that it's possible to make a cut using Cas9 and then knock in the cassette that you need to express your transgenic T cell receptor, shown here in green. And this effectively lets you knock out your endogenous T cell receptor, getting rid of the blue and purple. This is akin to what was going on in the previous slide. And the really important thing here is that instead of random integration of your transgenic cassette, you can knock it into an endogenous site, and this gives you really predictable and useful gene regulation of that locus. So the other challenge was the multiplexing that Carl June's clinical trial ran into. And this has been tackled by multiplexed base editing. So this was performed by Bo Weber when he was working in Brandon Moriarty's lab. And as you can see, delivery of a Cas9 RNP via electroporation was able to give really, really effective multiplexed knockouts. So 2/3 of the cells that they tried to engineer had 3 different genes knocked out. This is really, really impressive. And the added bonus of using a base editor is that base editors don't make double-stranded breaks. So this map shows all the different chromosomal rearrangements that are possible when you're making 3 cuts at the same time in a T cell that you're trying to engineer. And this plot sort of shows the frequency of chromosomal rearrangements. So when mRNA is used, it has a pretty substantial amount of chromosomal rearrangements going on. Using an RNP that cuts is a little bit less bad in terms of this genetic carnage. But when you switch from cutting enzymes to base editing enzymes, these chromosomal rearrangements almost entirely disappear. And again, those white pie charts that show 2/3 of the cells having all the knockouts you want, these were attained using base editors. So I think base editors have a huge value in creating knockouts safely without the risk of these genetic rearrangements. So DSBs notwithstanding, other types of cellular cures have made it to the clinic. There was a huge landmark case of patients like Victoria Gray who were dosed by CRISPR Therapeutics in, I think it was the summer of 2019. And she and other people are now cured of their hemoglobinopathies. So she was a sickle cell patient, and she's effectively cured. And this was done using a preformed Cas9 RNP. So really, really powerful technology. Unfortunately, the administration regimen is really, really risky and inaccessible and expensive. So this slide sort of summarizes all the severe adverse events that happened in a couple of patients that were dosed as part of this trial. And all of these side effects are associated with the route of delivery, not with the Cas9 itself. So this just highlights how much we need improved delivery technologies to get these enzymes into patients in vivo. So there's this big, big push to get away from ex vivo therapies, especially for stem cells where you require mobilization of the stem cells and conditioning. And there's a huge interest in developing in vivo targeted editing of stem cells and other cell types where you wouldn't need any mobilization of stem cells and you wouldn't want to do this therapeutic chemotherapy conditioning regimen, which can be really toxic. So later on, I'm going to talk about how my lab is working towards this ultimate goal. So briefly, I'll just tell you about some cases where RNPs have been delivered in vivo in preclinical work. And this slide sort of captures that size is a really big determinant of how a particle will be distributed in vivo. Because larger particles can't get through the endothelium, they can't get through tight junctions, size matters a lot. And I just want to highlight that there is a particle size sweet spot between 10 and 100 nanometers. And I think smaller is better when it comes to RNPs. And RNPs are the best in this regard because they're just 10 nanometers. So I should say it again, smaller is better when it comes to in vivo delivery. So I'm really enthusiastic about delivering RNPs that are not in a nanoparticle, but are just kind of individual particles. And one piece of research that did this is from the Gong lab. And they essentially took preformed RNPs and wrapped them in a polymer film. So they polymerized different functional groups around the Cas9 and created these, what I call shrink-wrapped RNPs, these nanocapsules. And these things are able to get into cells and perform endosomal escape and make their way to the nucleus. So these polymer wrapped RNPs were used for editing of the eye and of muscle tissue. So I think this is pretty encouraging. Another strategy was reported by David Liu and colleagues, where a preformed base editor was carried by a lipid nanoparticle, and this was used to edit postmitotic ear cells in a mouse. And this is really encouraging because they tried to do HDR, this is homologous recombination, the paste edit, knock-in using a separate DNA template, and they saw pretty much nothing. But when they used the base editor, used as a preformed RNP, they were able to see a good amount of cells getting edited in the postmitotic ear cells, essentially. And this is really important because knock-in using a template DNA relies on cell division, whereas a base editor doesn't rely on that. So that's another really appealing thing about using base editors in vivo. Paul McRae and colleagues were able to take preformed RNPs and complex them with amphiphilic peptides, and these were able to induce some editing in the surface of the lung. And finally, Dan Siewert lab has done some really cool things by finding different lipid nanoparticle compositions, which are able to steer packaged RNP nanoparticles to different organs specifically. So these can be sent to the liver or they can be sent to the lung, depending on the composition of the lipid nanoparticle. And in this reporter animal, you can see a little bit of fluorescence turning on in these various tissues. So it is possible to do systemic administration intravenously and have these nanoparticles steer their way to one organ type or another. So this is pretty encouraging. All right, so next up, I'm going to tell you about where we're headed, in particular, where my lab and our collaborators are trying to advance genome editing using delivery of engineered RNP complexes. So the big goal here is to take all the good things about Cas9, like its ability to bind a target sequence and its ability to cut that targeted DNA, and add new functions that will make it into an ideal delivery platform for use in vivo. And some of these functions are being able to navigate to a specific cell type, so add something like a molecular GPS. And then we also want to be able to open up cell membranes. So our corkscrew is going to stand in for that function. So the first project I'll tell you about very briefly is a collaboration with Alex Marson's lab at UCSF, and this is led by Wilson Lab postdoc Dana Foss and David Yuen, a clinical fellow in the Marson lab. And what we're trying to do on this project is take Cas9 and tether it to a targeting antibody that can recognize specific cell markers on cells of therapeutic interest, like T cells. So we in fact did this. We attached Cas9 to anti-CD3 antibody, and this recognizes a part of the T cell receptor. And this is really appealing because not only does it allow Cas9 to select for T cells as opposed to other cells, because the T cell receptor is specific to T cells, it also induces endocytosis into these cells. So sorry, this is our, of course, molecular GPS. It gives us targeting, but it also induces endocytosis. So to make sure that this first step was actually working, we tagged Cas9 with a small fluorophore and tracked its transit into T cells using microscopy. So when we use an anti-CD3 antibody and a tagged fluorescent Cas9, we're able to see punctate intracellular staining in T cells, which is consistent with endocytosis. And when we don't have that anti-CD3 targeting and we just use a sort of generic non-targeting IgG mix, we see mostly no staining, and if we do, it's sort of diffuse extracellular staining. So this suggests that we're able to target T cells and induce uptake into them with this antibody. Unfortunately, if we extract the genomic DNA from T cells like this, we don't see any editing. And that's because without a means of escaping the endosome, you wouldn't expect to see any editing. So to activate Cas9 for endosomal escape, we complex it with endosomolytic peptides. These are peptides that shouldn't be broadly cell penetrating, but they should give Cas9 the ability to escape the endosome. So this is our opener, our corkscrew, and fittingly, these peptides do tend to form helices, we think. And when we pair this with our T cell specific targeting, we can see really robust levels of editing. So as assayed by deep sequencing for CD4 knockout, we can see about 45% of alleles being knocked out when we combine the RNP with a peptide that is able to induce endosomal escape, as well as the T cell targeting antibody. So we still see some sort of background editing when we don't have that targeting antibody, but we see a huge boost when we induce that uptake. So this project is ongoing. We're constantly screening for new peptides and new strategies, and we're really excited about how much progress we made on this in the last year. So the next steps will be to take these peptides out of a kind of co-incubation context and actually attach them to the surface of the Cas9. We have two different ways to do this. We can either attach them to the surface of the Cas9 using some technology I'll tell you about in a few slides, or we can use adapter proteins that can bind to modifications in the guide RNA. So we are excited to apply this upgrade, and this will make a really nice platform that actually has a plug and play ability to target different cell types depending on which antibody we use. And so a thing that we're really, really excited about is sort of retargeting this platform to cells of therapeutic interest like hematopoietic stem cells. And this is really especially exciting because our institute, the IGI, has a clinical trial going on this year to do ex vivo correction of sickle cell disease, very similar to the CRISPR Therapeutics trial that I described earlier. But what we're doing is actually doing a knock-in. So we will be using template DNA to correct the beta globin locus. This is a little bit tricky to convert into an in vivo delivery strategy like my lab wants to do. So I think what we will likely consider are base editing type strategies where we just send a base editor and target it towards the stem cells. And this kind of strategy is good because the base editor is a sort of all-in-one approach. And this is something that we're really excited about figuring out in the coming years. So, this shift from ex vivo to in vivo, I won't belabor the point, but this is going to be huge, I think, in improving access to these therapies. Right now, an ex vivo cure is never going to cost it's never going to cost less than $1 million. So it's really, really going to limit how many people can get these cures. There are 100,000 people in the US with sickle cell disease, and we really want all of them to be able to afford a cure and access a cure. So Dana Carroll, genome editing pioneer, and I worked together on this perspective in the CRISPR journal that talks about some of these economic issues that I find really motivating. And I think that engineered enzymes could be a great way to more affordable cures because biologics have had such success. And if we turn CRISPR into another biologic, it'll be really momentous. All right, so in the last couple of minutes, I'm going to go really, really quickly through one more function we'd like to add to the CRISPR-Cas9 system, and that is to, embody the slippery outer coating of the Swiss Army knife and give Cas9 the ability to be a little bit slippery and improve its tissue distribution. So in some work from the Dowden lab a few years ago, they demonstrated that engineering a bunch of nuclear localization signals onto Cas9 actually gave it the ability to self-deliver into neurons in the mouse brain. And this is not too surprising because NLS sequences are very basic. They have lots of these lysines and arginines. And this sort of evokes a sequence like a cell-penetrating peptide, such as TAT, where there's lots of basic character. And so what this multi-NLS construct did is basically cover the Cas9 in these cell-penetrating peptide-like chemical groups. And this is great for cellular entry, but because membrane disruption is initiated by way of interaction with membranes, this propensity for membrane interaction could impede tissue distribution. So what my lab hypothesized as part of a collaboration with the Murthy lab is maybe we could make the surface of Cas9 slippery by temporarily attaching PEG molecules to that surface. And we did that using a chemistry that is called DEC, the DEC linker. And this allows conjugation of PEG onto the lysines that cover Cas9's surface. There's 125 of them. And the really, really cool thing about this is once Cas9 reaches the cytosol of a targeted cell, this linker will do a total reversal, and it falls off and restores all those lysines that are important for Cas9's function. So this is a bioreversible way to pegylate the surface of Cas9 that we've been working on. And we're very excited to see in this experiment that pegylation of the Cas9 surface did improve distribution throughout the mouse brain. So on the left is kind of two assays showing the same thing, the distribution of Cas9. This is another green fluorescent tagged Cas9 without PEG or with PEG. And on the bottom, we see some staining using an antibody that recognizes Cas9. And the distribution throughout the brain is much, much better when the Cas9 has been pegylated. And best of all, this pegylation does not hamper the ability of Cas9 to edit in the brain. And in this red on fluorescent reporter mouse, we do see improved editing once we've pegylated the surface of the Cas9. So this work has recently been published, at least a little bit of it, and we're working frantically to kind of maximize editing and brain distribution, because this is one of the really important things in moving from editing a small piece of anatomy like a mouse brain to a patient's brain, perhaps. So with that, I want to thank my brilliant and hardworking lab members, as well as our collaborators and those that provide us with funding to do this exciting science. And I'll be happy to take any questions.
Ross, awesome presentation and a great way to start things off. I'm certain I won't be the only one keeping a close eye on the developments coming out of your lab. To the audience, stick around again for the Q&A session that's going to begin in just a little bit. We're going to open that up for a panel discussion. So again, send in any question you might have for any of our speakers. Just click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit submit. Okay, let's get back to the presentations and turn things over to Max. Max, over to you.
Hi, I'm Max Selman. I'm the product manager for gene editing here at Aldevron. Really excited to talk with you today about our RNP manufacturing platform and the quality control assays we've developed to support GMP RNP manufacture. Just a little bit about Aldevron. We're a company, we've been around for over 20 years. We manufacture plasmid DNA, proteins, and mRNA from research grade to GMP. We're supplying companies worldwide, biotechnology companies, pharma companies, research companies. Our vision here is really to be the partner of choice in supplying these quality reagents to advance their research. We're very heavily involved in the cell and gene therapy space, as you can imagine, with Cas9 and RNPs. And so that's where I'm going to focus the rest of this discussion on. So by way of background, Aldevron, we were the first company in 2017 to release a GMP Cas9 nuclease as a catalog product. And what I'm showing you here on the screen are actually all the different nucleases Aldevron carries as catalog products. They have a common construct design, cleanly designed and simple. And the idea here is that you're going to have a very clean path from research grade to the clinic using these proteins. So the top two in particular, wild-type SP-Cas9 nuclease and then SPI-Fi-Cas9 nuclease, which is a high-fidelity variant manufactured in partnership with IDT. These nucleases we're actually taking to the clinic. And so we have these products off the shelf, research grade through GMP as catalog products. And the idea here is that you have a really consistent product design from when you're starting your research, you're doing your benchtop work, and then as you translate into the clinic, into your IND enabling studies and your actual phase 1 clinical trials, that you'll have a consistent supply of reagent that performs equivalently across those quality grades. And so we were the first company to do this back in 2017. And it's been really exciting for us to partner with companies and watch them grow from their small material requirements where they need not very much Cas protein, but as they've progressed in their trials and needed more material, we've been pushing ourselves to come up with new solutions that are going to continue to support these clients in their gene editing programs as they expand. And so what we've developed is the industry first GMP compliant manufacturing process for ribonucleoproteins, or RNPs. To support that platform, we've developed a number of new QC assays and adapted some others in order to support the GMP QC testing and release of these products. And so that's what I want to show you a little bit more about. So first, a little bit about the production workflow. It resembles in a lot of ways a traditional development through GMP workflow, in that you're going to start at our facility in Madison, Wisconsin, doing some research grade pilot batches. We want to confirm that the RNP performance that for the RNPs that Aldevron is manufacturing using those consistent research grade reagents is going to perform equivalently to the RNP that clients are making in their own hands. And so we'll use an Aldevron Cas9. We would complex it with a guide that's supplied by the client, a high quality guide, hopefully from a partner like Synthigo. And what we'll do is we'll complex that RNP and run it through our QC test panel. Now, it could be that the client has a good sense already for the complexing conditions that they want. It could be that Aldevron needs to help them develop those conditions and optimize that complexing. We can do all of that at research grade. Once we've aligned on research grade methodology and we've confirmed that the QC tests do work on the RNP, we're going to then go and do a transfer into our GMP facility. And then we're looking at a pretty traditional process from there, where you're doing product-specific validations on QC tests, you're doing engineering runs, and you're doing GMP manufacture and release. And so we're very excited to release our first GMP batch later this year, and to partner with more clients going forward on this workflow. As you can imagine from your own work handling Cas protein, the complexing of the RNP in a lot of cases is not the hard part. The challenging part is coming up with this QC test panel and then applying it in the context of GMP. So what we've done here is we've taken our Cas9 QC test panel and we've adapted it to RNP. So there's a few assays here that I'll focus in on. But in general, we're able to harness some of the tests, especially for contaminants that we were already using for Cas9, and then we're applying these customized assays to meet client requirements. So that would be a customized activity assay, and then tests to look at the content of free guide RNA that's left in the sample, and free Cas protein that's unbound, not bounded to an RNP in the sample. And so I'll just dial in on those two assays a little bit more here to with our last few minutes, just to show you what we're doing around free Cas and around free guide. So for free Cas9, we looked at a number of different conditions and ways of doing this assay. We looked at DSF, we looked at size exclusion chromatography. Ultimately, what we aligned on was an analytical cation exchange method that was able to resolve APO, or unbound Cas9, from RNP. And so we're able to apply this to RNPs not only as a release assay, but you can actually use this as a stability assay over time. And this is true for many of the assays on this panel. They would be stability indicating, we'd expect. And what you see here is that you've got clear RNP peak after 6 months at -80 storage. You actually still see that same peak at -22, but you do see some degradation at 4 degrees, and then near complete degradation of that RNP after 6 months at 30 degrees C. The take home here, number one, being that RNP should be stored at cold temperature, but number two, that the assay works and it's able to track stability as it relates to free Cas in this instance. For free guide RNA, we also looked at a number of different assays, assay methods, and we determined that just a simple native page assay was actually the way to go. And that's what we're doing for these initial clients is you run a native page gel, and you're able to quantify the guide RNA in that sample. And so, we're very excited to be able to apply these assays to client programs, and really to take this next step with clients. First as just the supplier of their Cas protein, but now actually supplying a ready-to-use gene editing reagent and being able to quality control it. We think that's going to be of huge value to clients, especially as you start treating more patients in a trial and you're feeling a desire to have that consistency and that quality control around the starting RNP. We're very excited to support this and look forward to further discussion and conversation. Thanks so much for your time. Really appreciate you listening.
Thanks, Max. Great presentation and really nice platform technology. So thanks for presenting that. To the audience, once again, Q&A session is going to begin right after this next presentation. So if you have any questions for any of our presenters, please send them in as soon as you can. So we're going to try to get to as many of your questions as possible. Again, just click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit submit. All right, let's get on to our final presentation. Kevin, I'm going to turn the mic over to you.
Hi, everyone. My name is Kevin Holden. I'm head of science at Synthigo, and it's my pleasure today to join the GEN virtual webinar on advancing direct enzyme delivery for therapeutic genome editing in vivo. So I would like to thank GEN, the organizers, for allowing Synthigo to be one of the sponsors, and it's my pleasure today to join Max from Aldevron and Ross Wilson from IGI as well to discuss this topic. So specifically, I'd like to talk to you about how Synthigo can help support therapeutic genome editing in vivo. And in particular, I'm going to be talking about the use of the CRISPR-Cas9 system for genome editing. So before I do that, I'd like to give a little bit of an overview on some of the applications of the CRISPR genome editing system, both in disease research and for therapeutics. So in disease research, the CRISPR-Cas9 genome editing reagents can be used to build models of disease. And so you can take these reagents as what we call an RNP, a ribonucleoprotein, and microinject them directly into a model organism like a mouse or a fruit fly or zebrafish to build an in vivo model of disease by knocking out a gene or building a mutation, for example. And then you can also transfect these reagents. We like to use nucleofection at Synthigo into immortalized cell lines, and you can do high throughput library screening for things like drug discovery. Also, you can transfect them into primary cells like iPSCs or T cells. And so by doing cell engineering, by making gene knockouts or making mutational variants, you can build these in vitro models of disease, which you can later then differentiate if you want to, or use to study a particular phenotype based on the genotype you've created. However, I would say, really one of the big promises of the CRISPR-Cas9 genome editing technology is the ability to do genome editing in the person, so gene or cell therapy. And so this can be done in one of two different ways. One is ex vivo gene therapies, where we isolate cells from a patient who has a genetic disease. And so this could, for example, be a blood stem cell, a hematopoietic stem cell. We do genome editing on those stem cells in the laboratory in vitro, and then we expand them and then ex vivo transplant them back into the patient to try to cure the disease. The other way is through in vivo gene editing, which is the main topic of this webinar today, whereby we take these genome editing reagents and we package them in some way, either in a vector or in some kind of nanoparticle or something, and then we apply them into the patient, and then hopefully they're directed to the correct site where genome editing can take place and cure genetic disease in a particular diseased organ. In this pictorial example here, the liver, for example. So let me talk to you first about how Synthigo can support CRISPR for disease research, and then I'll move into how we can support using CRISPR genome editing for in vivo therapeutic applications as well. So first of all, we can support this top section here, either building in vivo models of disease or transfecting into cells or doing high throughput library screening because as a company, we have a novel software-based RNA synthesis architecture that allows us to do mass production of the chemical part of the RNP, the sgRNA, the chemical guide RNA used for CRISPR. So these are chemically modified. They're synthetic guide RNA molecules. We synthesize thousands of these things every week. We can make them from very small scale to very large scale. And of course, we also offer these arrayed CRISPR gene knockout libraries that can be used for things like drug discovery or directly in assays. And now as a company, we also have full GMP production to enable gene therapies and clinical studies using guide RNAs for the CRISPR-Cas9 gene editing system. On the flip side, we also do a lot of cell engineering at Synthago. So we also are involved in building in vitro models of disease. So we have these automated high throughput platforms for doing cell engineering, whether that's in cell lines or iPSCs. And of course, we can do CRISPR gene knockouts and knock-ins like single nucleotide variants or gene tags. We have the ability to rapidly optimize transfection conditions for many different cell types, and we've done this now in over 350 cell types. And then, of course, we have systems to enable high throughput clonal isolation, imaging, tracking, and feeding of cells. And on average, we're generating hundreds of clones every week and shipping these out as projects to customers. Now, how can we support the in vivo and ex vivo CRISPR therapeutic world? From the ex vivo side, of course, we generate this research grade guide RNA material that people can use to do exploratory studies of gene editing in these relevant cell types like hematopoietic stem cells or iPSCs or T cells using this research grade guide RNA material. Of course, we also have the GMP version of this now as well, which allows people to go from discovery into the clinic. And then, of course, we can build, like I just mentioned, cell models. And in particular, we can engineer iPSC cell models, which can be very useful, or we can revert mutations to wild type. On the in vivo gene editing side, what we also offer is the research grade guide RNA that can be used for animal studies. So if people are packaging the RNP, or maybe they're mixing Cas9 mRNA with a guide RNA, they can package these and then they can put these directly into an animal because it's research grade. And then, of course, we have the GMP grade that people could use for clinical trials. But most importantly, I feel the way that Synthago can best support the in vivo CRISPR therapeutic community is through our research chemistry group. And then that's going to be the main focus of what I discussed today. Okay, so how can Synthago research chemistry support in vivo editing? So on the top left here, we have a standard N-modified guide RNA. This has these phosphothiorate and 2'-O-methyl modifications on the N parts of the guide RNA that provide superior CRISPR editing or base editing in virtually any cell type, because one of the reasons is they provide exonuclease resistance in the intracellular environment. More importantly, actually, they prevent these immune responses that can be caused by plasmids or guide RNAs that are derived through IVT, and there's been several publications now to show this. So these have been used for editing, as an example, like I mentioned, of the hematopoietic stem cells, and they're going to be used. They're actually being used currently, and also in upcoming CRISPR ex vivo clinical trials in a GMP format. Now, on the bottom here, we have what we call a heavily modified guide RNA, and so this is a molecule where there have been many additional modifications made throughout the backbone of the guide RNA. That's a 100 nucleotide sequence. And so this sequence here actually in particular was reported in Cell by research done from Intellia, where they used this heavily modified guide in conjunction with lipid nanoparticles to get very robust in vivo editing in a mouse. And they actually found that it was really important to utilize these heavy modifications on the guide RNA compared to just the N modifications. And there could be several reasons for that. The primary one is likely that it provides greater exonuclease resistance in the LNP or also the tissue environment. But they found in their study, at least, that delivery using this LNP, they could achieve up to 97% protein knockdown for at least 12 months inside of a mouse, which is pretty incredible. And so I just want to let you know that synthesis, the process to synthesize these molecules and develop them and actually make the material was done by the Synthigo Research Chemistry Group. So it's something that, that we help to enable for the study, and it's something that we continue to do. And so our research chemistry group is led by Dr. John Walker, who's the director of the research chemistry group. And the other work I'm showing you today has also been completed by Nastya Kadina, who's a senior scientist in chemistry research as well. And so some of our capabilities include not only the heavily modified guide RNA molecules, we can also do full modifications of the entire sequence as well. We can also make these chimeric RNA DNA hybrid molecules that can be used to bind specific regions of DNA, perhaps for some in vivo application. And importantly, I want to say that because we have this GMP capability now, we're able to go and carry researchers from the research grade that they're using in their lab for their experiments all the way into the clinic if necessary. Some other capabilities that we have that are quite interesting is we have the ability to work with RNA aptamer chemistry. And so we can conjugate these to the RNA molecules. We can synthesize these aptamers. And so what's interesting about these is that they form a three-dimensional structure that can be used to bind a specific target, for example, inside of a cell or to a particular cell type even. So that's a really critical part of working with aptamers. We can also enable the use of other genome editing technologies, including things like prime editing, where you need much longer guide RNAs, or to work with other CRISPR nucleases that may require different lengths of the guide RNA. So we're actually capable of synthesizing these molecules up to 160 nucleotides quite frequently now. We're also able to do many different types of modifications besides the 2'-O-methyls that I already mentioned to you. We can use these fluoro modifications. We can generate A-base6 sites as well, and we can also insert these linkers. And so being able to utilize all of these different moieties on the sugar backbone of the RNA molecule can bring certain advantages when doing editing in different situations, whether it be editing in vitro in a cell line or in vivo in an animal. We're also capable of labeling the RNA molecules as well. So this can be done through labels like biotin or thiol or amines. We can also do fluorescent labeling, and we've done quite a few of these projects now where we've labeled using these different dyes in different wavelengths. So these can actually also be important for visualizing where RNP complexes can go in vivo. We also work on novel chemistry development. This is a system that we devised in-house that we call CRISPR-off guide RNA. This is a system where we actually use different chemical linkages in the molecule that are susceptible to breakage when exposed to light. And so compared to a normal guide RNA, when you shine light on these, and it can be inside of a cell, these guide RNAs will break up and the activity of the RNP is completely lost. And so we showed in a very nice publication in Nature Communications at the end of 2020 that you could use this approach actually to reduce the amount of off-target at known off-target sites. And so this type of temporal control is capable because the on-target editing typically occurs much faster than the off-target editing. And so we can allow the on-target editing to happen. We can shine light onto the cells, and then the guide RNAs will be broken up and the off-targeting does not have time to actually catch up. And so in the paper, we actually showed across quite a few sites here that we could alter the on to off target editing ratios based on our ability to inactivate these guide RNAs. And we can also enact spatial control as well. As you can see in the image here on the right, in this GFP expressing strain, we can actually use a filter to hit cells that have these guide RNAs in them and actually create patterns, for example. So we're currently exploring how this technology can be utilized both for perhaps in vivo and ex vivo CRISPR-based therapeutics as a way to reduce potential off targets. Be interesting to see if it could maybe one day put these into an animal or a patient and shine light through the skin and see if you can deactivate them. Let's see. Our research chemistry group is also involved in some nice in vivo genome editing collaborations as well. We're currently involved in several of these as a way to utilize our capabilities to improve or enable in vivo genome editing. We have a very nice collaboration with Gerald Schwenk's lab at the University of Zurich. Here's a recent paper by his lab group showing how they used cytidine base editing in hepatocytes in vivo to get some very nice editing results. Our collaboration is still ongoing, so I can't tell you exactly what it's about, but you could kind of guess that it might be along that path. Also here is Stanley Key from Stanford University, and recently they put out a paper on actually utilizing the Cas13 CRISPR system to act as an antiviral strategy to fight SARS-CoV-2. So that's a little bit in line with the collaboration that we have going on with them as well. And also, I'd like to mention Ross Wilson from the Wilson Lab at IGI. We've done some RNA work with them as well, but I'll let Ross talk about what they're working on specifically with regard to chemistry on guides or aptamers and or maybe chemistry on proteins as well for their in vivo editing and targeting. Okay, so that was really brief, but I just wanted to give a little overview of the way that Synthigo can support therapeutic genome editing in vivo, mostly through our research chemistry capabilities. Again, we can also help to enable researchers by building cell models in iPSCs or cell lines, and also because we have a GMP compliant factory now, we can support researchers who are trying to do genome editing clinical trials to go all the way from discovery into the clinic as well. So if you have any questions about anything I've said today, feel free to ask me any questions in the Q&A session, or you can reach out to my email down here, or you can reach out to Synthigo in general at synthigo.com/contact. So, all right, thank you for your time, and I will let the virtual webinar with Jen continue.
Thanks, Kevin. Great presentation and data, and a really nice way to wrap up a great webinar. Thanks for that. For the audience, our Q&A is going to start in just a few minutes. So again, as I've said throughout the whole presentation, if you have any questions for our three presenters, don't hesitate to send them in. Again, just type the question into the Ask a Question tab on the right-hand side of your screen and hit submit. All right, I want to thank those audience members that have sent questions in already, but for any of you thinking about it, don't hesitate, send them in, and bear with us for just a moment as we transfer into the Q&A session.
All right, everyone, thanks for joining us for the Q&A session. We have a bunch of questions, so let's get to them in just a minute. I did want to point your attention to the resources section there. There's a bunch of stuff in there from the Aldebaran team and Synthigo team. I did forget one, guys, and I'm going to post it back in. My apologies for that. There was a post from the Synthigo team that I'll put back in there, a CRISPR benchmark report. So make sure you check back in and get that report and download that. It's a pretty interesting one. So let's get to the first question. First one's going to be for Ross. Ross, one of our audience members wants to start out by thanking you for a great presentation. And they wondered how the status for the delivery of prime editors is going at the moment.
Hey, thanks a lot. And also I want to thank everyone for attending. This has been a really great session, so I appreciate everyone's attendance. I'm extremely excited about prime editors in theory. Currently, we're still waiting to see if they can really make the leap from, a promising research tool into a really useful one. I have heard a few promising reports about prime editors being used as RNPs, which I think is very exciting because, as I tried to make the case, delivering an RNP is useful for a lot of reasons. And if you can do all the things a prime editor can do, so make precise changes that are not hindered by the limitations of base editors, this could be a massively useful therapeutic approach. So I think we're not quite there yet, but there's a lot of hope and excitement, and I think the next year will really tell us how rapidly these are going to be moving from preclinical work into the clinic.
Great, thanks, Russ. As a follow-up to that, one audience member would like to know, how hard is it for this technology to reach clinical trials?
Well, it really depends on how novel your delivery strategy is going to be. So I think it's absolutely remarkable that it's only been 7 years between the sort of announcement of CRISPR-Cas9 as a potential genome editing tool until in 2019, people were being dosed with Cas9 RNPs. So I think 7 years is a very fast translational window for a totally new platform. But again, in this case, the delivery problem was sort of skipped because these are ex vivo trials and the cells are being removed from the patient. So that rapid timeline got to rely on decades of transplantation research. So we have a little distance left to go before RNPs are going to be used in vivo in the clinic, but I think it's conceivable that in 5 years, clinical trials could be underway for clinical delivery of RNPs, and certainly non-viral delivery is already in the clinic. As Kevin alluded to, Intellia is dosing people with lipid nanoparticles carrying mRNA, and so the timeline can be quite quick for genome editing in general. So I think this is a new landscape entirely, which is very thrilling, I think.
All right, thank you, Ross. Max, we have a question for you. An audience member would like to know, what is the smallest amount of RNP that Aldevron can manufacture research grade and cost effectively?
Sure, that's a great question. And just briefly to echo Ross and thanking everyone for joining us today. RNP manufacturing, we can make pretty small quantities. As you probably know, it's more or less the complexing is just mixing component A and component B, and we have a methodology for doing that that can be scaled down to quite small. Now, in terms of actually implementing the QC methodologies that I talked about, you probably do need a few milligrams of complex material in order to actually do the development and implementation that's necessary. So I'd say minimally, let's say single digit milligrams. It scales much larger. Our first clinical batch is we're going to do clinical lots at 500 milligram scale GMP. And so I would say the complexing range is quite broad, but specifically implementing the QC. Once you've implemented the QC methods, it's just a matter of making a batch to the scale that you require.
All right, thank you for that, Max. Ross, back to you. An audience member says that while it's lovely to be able to use an antibody to deliver Cas9, how do you deliver a protein complex that is sure to be antigenic?
Yeah, that's a great question and something that we're constantly thinking about and aware of. So, as many people appreciate, the most popular versions of Cas9 are derived from two human pathogens, Staphylococcus aureus and Streptococcus pyogenes, and these are pathogens that may have already caused patients, to have antibodies against the corresponding Cas9 proteins. And even if someone doesn't have these yet, they would be likely to develop them after a therapeutic dose of Cas9, for example. So there's sort of two ways that we think about this. One way is just optimism. If a patient's titers of antibodies are low for Cas9, then it might be partially neutralizing against some of the dose, but it shouldn't cause a massive immune reaction for a single dose. And one reality here could be that any genome editing therapy could be limited to a single dose if it is immunogenic. And so fortunately for genome editing, one and done is an option, so that's sort of what we strive for. Another way to think about this is to use some of that reversible pegylation technology that I described in the last part of my talk, where the Cas9 can be coated in a shell of polyethylene glycol, which can provide some shielding from recognition by antibodies and other immune components. And this shielding would essentially fall off of the Cas9 once it's inside the cell. So it would give you protection from immune response only in the context where you really need it. And one thing I really want to highlight about non-viral delivery is that if you don't have persisting expression of your enzyme, your cell-mediated immune responses will be much milder. So I think one of the reasons we don't see a lot of viral delivery of CRISPR enzymes in the clinic right now is that outside of immune-privileged places like the brain or the eye, you could expect the immune system to clear out any cell that is expressing a CRISPR enzyme in a long-term fashion. So viral delivery has a major disadvantage in this regard, and that's something that we're hoping to take advantage of in performing RNP-based delivery.
All right, thank you, Ross. Another question for you that I think is pretty good. Will genome editing therapeutics make the leap from rare diseases to more prevalent conditions, and what might determine the timing of this transition?
Yeah, that's a great question. One of the things that is really, really gating here is the safety profile of these genome editing strategies. So recently, many people may have heard the news that Bluebird's lentiglobin trial to treat hemoglobinopathies was put on hold because there were some cancer and pre-cancer reports in some of the patients. So this sort of tragic news is upsetting, and it's also one of those things where clinical trials are currently sort of limited to rare disease because the safety profile of these next-gen therapeutics is not totally understood. So I'll emphasize that this was an ex vivo trial that uses aggressive chemotherapy and a lentivirus. A lot of the things that I would not personally imagine for the future of genome editing therapeutics. So I guess the really important gating thing here is safety. So because this is new territory, I think it's likely that in the next 5 or 10 years, people will mostly be focusing on rare disease simply because that's a case where it can be justified to use something that we have some questions about, right, or some concerns. But at the same time, I'm very optimistic that genome editing can be done safely for a huge number of situations. And I think that the safety profile is going to be very encouraging and very easy to characterize. So I think that once we have a little bit of trust in the technology over the next few years, it really does have the potential to make the leap from rare disease to widespread disease. And one specific example of this is Verve Therapeutics, who is using base editing, which I think is going to have a very nice safety profile, to treat liver disease that has a rare disease manifestation, but one could also imagine these similar genome editing strategies of the liver to help people, control their cholesterol levels in the same way that many, many people currently use statins to do. So it's kind of a very long-winded answer to say that I think in time, once we're very confident about safety, I have no doubts that genome editing is going to make the leap to kind of more prevalent disease to give people, one and done options for cures instead of sort of lifetime of taking a pill or managing a disease.
All right, thank you, Ross. I very much like your optimism. I hope you're right. Kevin, we have a question for you. One of our audience members would like to know, why might guide modifications be more or less important depending on if Cas9 protein or Cas9 mRNA is delivered in vivo?
Yeah, it's a great question. And again, also just want to echo Max and Ross. Thanks for having me, and a great set of presentations today. Nice to see you guys again. Also, I guess just echoing on one of the questions Ross just answered about cutting down on the immunogenicity of potentially a Cas protein could be one way to negate that, can be to deliver mRNA in vivo. And so, actually right now we've got mRNA vaccines, right, for COVID thing, which is a great thing. So, and that's actually to carry on to the question that was asked, which is why the guide modifications might be more or less important. And essentially it's because if you're delivering the mRNA, it's going to take a while before the cell starts to make that. So it's going to be really important for that guide to maintain stability while the mRNA is being translated. So, at least in the studies that we've seen so far, using the heavily modified versions of the guides for in vivo delivery is absolutely critical for them sticking around and actually for the editing to be effective. So that was showed in the paper that Intellia published, and we've also seen it in other studies that we've been involved with.
All right, thank you very much, Kevin. Ross, back to you. Looks like we have time for one more question. I think this is a good one. It's a nice, thought-provoking question to end on. An audience member says, in your presentation, you talk about how gene therapies tend to cost at least $1 million per cure. How will this tradition impact the widespread adoption of clinical genome editing?
So, obviously, $1 million cures are life-saving and life-changing for people with life-threatening rare diseases. And, I don't want to downplay the value of these things that are now accessible. And, many people never thought we'd see a lot of these technologies. So, it's wonderful that, these next-generation therapies are being approved, but it causes a little bit of a potential bubble because, the promise of genome editing is that it can theoretically treat lots and lots of diseases. And if you try to imagine each of these cures costing $1 million, I think there's going to be a problem. And certainly it won't be amenable to wider distribution, rollout, whatever you want to call it, adoption. So I think that there's an imperative need for these costs to come down. And I think one of the ways that can happen is through what we've talked about today. So CRISPR is great because it's a platform technology. It's sort of plug and play regarding which part of the genome you're going to manipulate. If you can find a way, if someone can find a way to create sort of plug and play delivery technologies, so technologies that can target different parts of the body, or even allow you to selectively treat multiple diseases of any given organ, then it's easy to imagine that the clinical investment required in, determining safety and efficacy of each of these platforms, that clinical investment should start to shrink. And I think that's one of the things that causes these rare diseases to be so expensive. Each one of these new therapeutics has to be totally explored in the clinic, and that's very expensive, and the number of patients that can be treated is pretty small. So for a company to recoup their investment, the price has to be high. But as we start to create plug-and-play cures, hopefully the clinical investment will start to be smaller because these things can be de-risked in different ways without performing, a massive full-size clinical trial. Again, once we start to trust some of our delivery technologies. So that's where I think we're heading. And, something that's readily manufactured like an RNP is really going to pave the way to this sort of future. And I think it's going to be hard to get there with viral manufacture just because this hasn't become much more robust in recent years. So I think delivery and use of non-viral delivery technologies is really going to help change the accessibility of these cures.
All right. Thanks very much, Ross. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on the GEN website at genenginews.com for up to a year. So if you missed any parts of it, You can watch it again, or feel free to forward that link to any of your friends and colleagues, which we always recommend. And like I said earlier, check out that resource section, and don't forget to come back in a little bit and check out some of the new additions that I'm going to put in there soon. I'd like to thank Ross, Max, and Kevin, again, for their very informative presentations, and I'd like to thank you, the audience, for your attention and thoughtful questions. And a very special thanks to Aldevron and Cithico for sponsoring this webinar. Hopefully, we'll see you again at another Gen webinar in the near future. Goodbye for now. Everyone stay safe and healthy.